Did you know that there's a whole field of study related to nutrition in agriculture? Don't you worry, you don't have to be an expert to manage a system. Let's go! Guys, are you still with me? This is a generalized model of nutrient cycling in a terrestrial ecosystem. As plants take up nutrients from the soil or water, they become incorporated into living tissues or organic matter. As the tissue senesce, the dead organic matter returns to the soil or sediment surface. Various decomposers transform the organic nutrients into mineral form, and they are once again available for uptake by plants. This process is called internal cycling or nutrient cycling. What are nutrient cycling? Nutrient cycling is the continuous exchange of nutrients or elements among organisms and between organisms and their physical environment. It is also known as biogeochemical cycle. There are two types of nutrient cycle, the gaseous and sedimentary. The source of gaseous nutrient cycle is present in the atmosphere. Examples of this cycle are carbon and nitrogen cycle. The source of sedimentary nutrient cycle is present in the crust of the earth. Examples of this cycle are phosphorus and potassium cycle. The carbon cycle. Carbon is one of the elements that cycle through nature. During photosynthesis, plants combine carbon dioxide from the air and hydrogen from the water to make carbohydrates. Some of these carbohydrates are stored in the tissue of the plant. Others are used by the plant for energy. When the plant is eaten by an animal, the cells of the animal break down the plant's tissue during digestion. These release the stored carbon and other nutrients into the animal system. As the animal breathes out, carbon dioxide is released into the air and the cycle can begin again.
The nutrient cycle describes the use, movement, and recycling of nutrients in the environment. Valuable elements such as carbon, oxygen, hydrogen, phosphorus, and nitrogen are essential to life and must be recycled in order for organisms to exist. Nutrient cycles are inclusive of both living and non-living components and involve biological, geological, and chemical processes. Carbon is essential to all life as it is the main constituent of living organisms. Carbon cycles through the environment is as follows. Carbon dioxide is removed from the atmosphere by photosynthetic organisms such as plants and cyanobacteria, and used to generate organic molecules and build biological mass. Animals consume the photosynthetic organisms and acquire the carbon stored within the producers. Carbon dioxide is returned to the atmosphere via respiration in all living organisms. Decomposers break down the dead and decaying organic matter and release carbon dioxide. Some carbon dioxide is returned to the atmosphere via the burning of organic matter or forest fires. Carbon dioxide is trapped in the rock or fossil fuels can be returned to the atmosphere via erosion, volcanic eruptions, or fossil fuel combustion. Similar to carbon, nitrogen is a necessary component of biological molecules. Some of these molecules include amino acids and nucleic acids. Although nitrogen is abundant in the atmosphere, most living organisms cannot use nitrogen in this form to synthesize organic compounds. Atmospheric nitrogen must first be fixed or converted to ammonia by certain bacteria. Nitrogen cycles through the environment is as follows. Atmospheric nitrogen is converted to ammonia by nitrogen-fixing bacteria in both aquatic and soil environments. These organisms use nitrogen to synthesize the biological molecules they need to survive. Ammonia is then converted to nitrite and nitrate by bacteria known as nitrifying bacteria. Plants obtain nitrogen from the soil by absorbing ammonium and nitrate through their roots. Nitrate and ammonium are used to produce organic compounds. Nitrogen in its organic form is obtained by animals when they consume plants or another animals. Decomposers return ammonia to the soil by decomposing solid waste and dead or decaying matter. Nitrifying bacteria convert ammonia to nitrite and nitrate, while the nitrifying bacteria convert nitrite and nitrate to nitrogen gas, releasing nitrogen gas back to the atmosphere.
In this experiment, a known quantity of dried leaves is placed in a mesh bag on the forest floor. Bags are retrieved at various intervals, and the mass lost due to consumption by decomposers is tracked through time.
This is a diagram illustrating the exchanges between liter bag and the soil in a standard liter bag experiment used to quantify the process of decomposition. Decomposer organisms like bacteria and fungi colonize the plant litter. As litter is consumed, a significant proportion of carbon is respired, and nutrients bound in organic matter are mineralized and released to the soil. To convert plant carbon to microbial biomass, mineral nutrients are taken up in the process of immobilization. The difference in the rates of mineral nutrient release and immobilization is the rate of mineralization. Note that as time progresses, the residual organic matter in the litter bag is composed of a growing proportion of microbial biomass as the original plant material is consumed, respired, and converted into microbial. The process of breakdown of complex organic matter into inorganic substances like carbon dioxide, water, and nutrients is called decomposition. The organisms involved in this process are called decomposers. The most common decomposers in an ecosystem are fungi, bacteria, some algae, and few protozoans. Decomposition is largely an oxygen-requiring process. The rate of decomposition is controlled by chemical composition of detritus and climatic factors. The process of breaking down a substance into its constituent parts is called decomposition. It is brought about the reducer organisms. These organisms break up the dead organic matter into a small organic molecules which they can utilize themselves as nutrients or inorganic compounds that are released into the environment. The rate of decomposition of organic compounds varies. For example, proteins, fats, and majority of carbohydrates decompose rapidly, and cellulose, chitin, lignins decompose slowly. The upper layer of soil is the principal site for decomposition processes in terrestrial ecosystem, while in aquatic ecosystem, the bottom portion of the body is the site for decomposition.
Decomposition is the breakdown of chemical bonds formed during the construction of plant and animal tissues. Decomposition involves an array of processes including leaching, fragmentation, digestion, and excretion. An essential function of decomposers is the release of organically bound nutrients into an inorganic form. The wide variety of organisms involved in decomposition are classified into groups based on function and size. The microflora, comprising the bacteria and fungi, are the group most commonly associated with decomposition. Bacteria can be grouped as either aerobic or anaerobic based on their requirement of oxygen to carry out respiration. Invertebrate detritivores are classified based on body size. Microbivores, fetal bacteria, and fungi.
Microbial decomposers use carbon compounds contained in dead organic matter as the source of energy. Various carbon compounds differ in their quality as an energy source for decomposers. Glucose and other simple sugars are easily broken down and provide a high-quality source of carbon. Cellulose and hemicellulose are intermediate in quality, whereas the lignins are of very low quality and therefore decompose the slowest. The quality of dead organic matter as a food source for decomposers is influenced by the types of carbon compounds present and by nutrient content. Because of the low quality of lignins as an energy source for decomposers, there is an inverse relationship between lignin content of plant litter and its decomposition rate.
The physical environment directly influences both macro and micro decomposers. Temperature and moisture greatly influence microbial activity. Low temperature and moisture inhibit microbial activity. As a result, the highest decomposition rate occurs in warm, wet conditions. The influence of temperature and moisture results in geographic variation in rates of decomposition that relate directly to climate.
The process of decomposition continues as the litter degrades into a dark brown or black homogeneous organic matter known as humus. As this organic matter becomes embedded in the soil matrix, it is referred to as the soil organic matter. Although high in nitrogen, humus decomposes very slowly due to the lack of quality of carbon compounds. Despite the slow rate of decomposition due to its sheer abundance, the decomposition of humus represents a significant portion of the carbon and nutrients released from the soils. There is an active zone of root growth and depth, characterized by an intense microbial and fungal activity. Decomposition in the rhizosphere is more rapid than in the bulk soil because plant roots use carbon-rich exudates to supplement the decomposition process of low-carbon quality organic matter. Nutrients immobilized in bacterial biomass are then released to the soil as microbivores that feed on the bacteria. Energy-rich carbon exudates from the root tips function to supplement the energy demands of bacteria and fungi in the rhizosphere. The bacteria then meet their nutrient demands by breaking down the soil organic matter that is rich in nitrogen but of low carbon quality. The result is a high degree of immobilization of nitrogen from the soil organic matter into bacterial and fungal biomass.
One strategy from indicating climate change is to increase the amount of carbon stored in the soil in the form of soil organic matter or SOM. Of particular interest is stable SOM, which can store carbon for centuries before it can return to the atmosphere as carbon dioxide. Stable SOM forms from decomposing plant matter, also called litter. To better understand this process, the decomposition of tall grass litter was investigated in a recent study. The findings reveals two distinct decomposition pathways which result in SOM with different degrees of stabilization. SOM primarily consists of decomposed plant matter, the microbes involved in its decomposition, and their waste. Plant litter consists of the slow decomposing fraction which includes many of the structural components of the plant such as lignin, and the fast decaying water-soluble fraction which includes many of the non-structural components such as sugars and amino acids. It has long been assumed that stable SOM is almost exclusively formed from the slow decaying plant litter component. However, monthly evidence suggests that water-soluble litter component, which can be efficiently transformed by microbes, can also form stable SOM, where the stability is provided by binding with minerals. Isotope labeling was used to track the tall grass plant litter during decomposition. The researchers fed heavy carbon and nitrogen isotopes to the plants as they grew, and added the labeled plant litters to the soil in a natural tall grass prairie. These labels allowed the researchers to follow the plant matter as it descended from the soil to become incorporated into microbes. They could then identify the litter degradation products as they formed new SOM.
Nutrients are sequestered during microbial growth and will remain locked up in fungal and bacterial biomass if consumption by protozoa and nematodes did not constantly remobilize essential nutrients for plant acting. Due to relatively small differences in carbon and nitrogen between predators and prey, and a relatively low assimilation efficiency, only 10–40% and 50–70% of prey carbon will be used for biomass, production of protozoa and nematodes respectively. The excess nitrogen is excreted as ammonia and hence readily available for uptake by plants.
Composition in aquatic ecosystems varies as a function of water depth and flow rate. In open water environments, dead organisms and other organic matter, called particulate organic matter, drift downward to the bottom. On its way, the particulate organic matter is constantly being ingested, digested, and mineralized until much of the organic matter is in form of unique compounds by the time it reaches the bottom of sediments. Bacteria decompose organic matter on the bottom sediments using aerobic or anaerobic respiration depending on the supply of oxygen. Dissolved organic matter in the water column also provides a source of carbon for decomposers.